as an organizer, it was pretty easy for me to get a talk because they couldn't say no when we submitted an abstract. And I made sure that those people who should be giving the talk are actually not here. So I finally get to talk again. No, seriously, the people who actually did the work, they have moved on to better and more. And so I'm happy to be here today. I also want to say that as a biologist, I've been coming to this meeting for many, many years. And I'm constantly amazed by the work that is presented here, the kinds of tools and algorithms that you generate. And I, as a biologist, cannot do what you do, but the tools that you provide are so important for us, and you enable us to do the kind of work that you're going to get a snapshot about today. And so I'm going to stick with the theme of cancer, but this time with a nutritional angle. So you might have heard of the dependence of cancer cells on high energy or other metabolites. And so the most important one that you probably know about is the Warburg effect, which is basically the cancer cells need a lot of ATP and glucose and energy. A lesser known dependency, metabolic dependency, is referred to as the Hoffman effect. And that refers to the dependency of cancers on the presence of methionine. So methionine, why methionine? Methionine is an essential amino acid that is taken in by diet. And in this methionine cycle, methionine is used is converted into SAM, and SAM is used as a cofactor to methylate a bunch of different aspects of biology. So it's either proteins, it's RNA, it's DNA. And so the input here through the diet is being taken up in many versions of the cell. And what cancer cells, what is known is that cancer cells, they're happy to grow in the presence of methionine, just as normal cells are. But if you start to starve the cells of methionine, and you give them the precursor of methionine, which is homocysteine instead, they stop growing. They stop growing and they are not able to proliferate at the same rate as they usually would be. And this is not because cancer cells cannot convert homocysteine into methionine. It is simply that the demand of the cancer cell to proliferate requires a much higher concentration of methionine. So this project started with my collaborator, Peter Kaiser, who has been investigating this Hoffman effect for quite a while. And so what we wanted to do is to recapitulate and sort of see whether the earliest observations can be seen again. And that is when you actually take animals, mice here, and you put in a xenograft tumor and then either feed them control or a low methionine diet, what you can observe is that the tumor volume for those mice that only receive the methionine, low methionine diet is much lower than for the control mice, even though the weight of these mice do not change. So they get enough energy, but somehow the tumor doesn't grow. So what do we do from this point? How do we study this? So of course we go into cell lines. When you take many different cancer cell lines, here's a series of breast cancer cell lines, but you can take colon cancer cell lines, you can take glioblastoma cell lines, et cetera, et cetera. And if you grow them in a dish, either in the presence of methionine or at starvation conditions, what you can see is that in the presence of methionine, you get a proliferation, whereas in the absence of methionine, they do not proliferate as well. So this is a nice system that allows us to investigate the potential mechanisms that sort of underlie the Hoffman effect. And what we wanted to do is we wanted to take one of these cell lines and we wanted to see whether we can generate a methionine resistant cancer cell line such that we could have a cell culture system where we have genetically very similar cell lines to compare to. And you can actually do that. So through colony growth, we can generate a series of methionine resistant cell lines. And what you can see here, and when we feed those resistant cell lines, or when we let them grow in methionine restricted media, they grow just as fine as normal cells do. What happens though to these converted cancer cell lines is that they no longer act as cancer cell lines. So through the loss, whatever they lost, through the loss of this methionine dependency, they lost their cancer phenotype. Okay, so now we have a very nice system in place where we can start to do transcriptomics, where we basically compare the wild type cancer cell line, the 468 cell line, with our derived methionine resistant or methionine stress resistant cell line, the R8. So transcriptomics give us a lot of hits. We've done these in time series where we take gene expression profiles, RNA-seq, at either 30 minutes or in this case 12 hours. I'm just going to show you the 12 hours. This is early on in this proliferation pathway. So what we try to evaluate or get information on is really what happens early on. And at the transcription level, there are many genes that are upregulated. There are many genes that are downregulated. When you compare the differences between the two cell lines, you find that there are pathways targeted that they deal with fatty acid synthesis. And basically, this is a story that I can't tell you due to time, but there's a whole rearrangement of the cellular architecture of these cell lines as they become resistant to methionine stress. Now, I wouldn't be here if we wouldn't be doing RNA-seq analysis and alternative splicing. And when you carry out the RNA splicing analysis, what you see immediately is that the cell line that is affected by the methionine stress undergoes a huge amount of alternative splicing. This is all kinds of alternative splicing. And we're typically interested in how do you get to these changes. So when you look at the volcano plot here, what you see is that there is a significant increase in the loss of exon inclusion. So putting these cells under methionine stress, somehow you start to lose exon inclusion. So that seems to suggest that, well, there's something going on there within the spliceosome or within the system, and now the spliceosome is no longer able to recognize certain exons that it usually does. We can evaluate a few features. We can evaluate genes that are affected by alternative splicing, and wonderful enough, there are pathways that relate to cell cycle control. And when we look at three different features, one are splice site strength to the left. In the middle, we have a feature that looks at the conservation of the exon length, which is actually very important, and it's an evolutionary feature. Or if you look at sequence conservation, what you can see is that these exons that we're losing upon methionine stress, they are not such good exons. And what we would almost say is that they are likely younger exons. So these are exons, we're losing exons that do not have the highest recognition potential. And so this sounds a little bit like SMA in some ways, but the model that comes out here is that somehow methionine stress reduces the efficiency of splicing. So to test that directly, we used a reporter where basically a luciferase expression vector is either is interrupted by an intron. And so the idea here is if methionine stress reduces the efficiency of splicing, then what we would expect to see is that there would be less luciferase generated upon methionine stress, which we do observe then within the context of the methionine dependent cell lines, but not so within the context of those that are resistant. Okay, what mechanisms could be in place here? And so here I'm doing the reverse of what everyone else is doing. I'm trying to find the needle in the haystack, whereas most of your tools generate the haystack, okay? So we stared at many of the genes that are differentially spliced, differentially expressed, and we ended up focusing on one, and that is PRMT5. So PRMT5 is a protein arginine methyltransferase. Well, there you go. That's the first clue. We add in or we reduce methionine. Of course, that has something to do with methylation. And what we also know is that PRMT5 has multiple functions within the cell. So it does affect histone acetylation, methylation. It does play a role somehow in splicing. In fact, it is very well known to be the methylase that methylates the SM proteins that make up the ring, that make up the SNRPs. So there is a direct potential connection between PRMT5 and splicing. And the way the SNRP assembly works is that the SM, there are 7 SM proteins that form a ring, as shown here. They get incorporated into the SMN complex, which is a chaperone complex that assembles the whole SNRPs. So all of the SNRPs go through this pathway. And so it is possible that losing methylation of these SM rings would then reduce the efficiency of SNRP assembly, and therefore you decrease the effective concentration potentially of SNRPs, and therefore you reduce splicing efficiency, overall splicing efficiency. Let me talk a little bit more about PRMT5 because it's also often overexpressed or differentially expressed in cancers, and it is actually a target that is being evaluated in preclinical trials. So there are inhibitors out there. However, those inhibitors, as we just heard from Mathieu, the holy grail of cancer therapy is you got to target the cancer cell and not kill the normal cell. And just the inhibitor that is evaluated right now cannot do that because it doesn't differentiate between a cancer cell and a normal cell. And the last clue that we got was that there is actually an alternative splicing event within PRMT5. It is an intron retention event. So here we have a potential needle that is in the haystack. And so what we ended up doing was to evaluate, do these SM proteins get differentially methylated? And so what you see here on the left-hand side, and I'm pointing here to the SMD1, upon growth in the restricted medium, you see a loss of methylation of the SMD1. You also see a loss of interaction of SMD1 with the SMN complex. That is quantitated here. So the data shows that methionine stress somehow interferes with the efficiency of PRMT5 in the context of the cancer cells, and only in the cancer cells because the resistant cells do not do that. So this methionine diet specifically targets the methylation of the SM rings within the context of the cancer cells. So now the question is, how can we use this information? And we know that there are that there are inhibitors out there. So is it possible that we can use this idea of nutritional dieting and in combination with the inhibitors to sensitize cancer cells towards the inhibitor? What you see on this slide is you see the growth effect of the methionine-dependent cancer cell line with an increase in concentrations of the PRMT5 inhibitor at various levels of methionine within the cell culture medium. And you compare that to the methionine-resistant cell line, which is down here. And basically, what we can show here is that the methionine restriction and PRMT5 inhibition synergizes. They work within the same pathway, and they can selectively inhibit cancer cells over the methionine stress resistant or normal cell line. So that's pretty much the story I was going to tell you in terms of the summary. We have methionine restriction, alters transcript level and mRNA levels. That's nothing surprising there, because if you stare hard at your tissue culture cell line, you know it's going to change splicing, and so you will always see differences in splicing. But it's not always the weaker exons that are preferentially targeted. We know that this methionine restriction has an effect on the methylation pattern of SMD1. So what we believe is happening is that you have a reduction in SNRP assembly and a reduction in the effective concentration of SNRPs, and therefore lower splicing efficiency. And therefore, the weaker exons are the first ones to go. And we believe that that the methionine restriction is within the same pathway of PRMT5. So our future studies and how can we use that information is attempting to look at combination therapy, where first steps in the context of mice, we will evaluate whether methionine restriction and the administration of the PRMT5 inhibitor will selectively reduce the cancer burden. And so with that, I want to thank the two who did the most of the work, and that's Francisco and Stacy. I want to thank Peter Kaiser, whose lab is really responsible to set the foundation of this work. And I want to thank you for your attention.
Any questions? Thank you so much for the talk. So you have seen that the methionine stress reduces splicing fidelity. So what about other type of cellular stresses? Do you have studied them, or you think that's equivalent mechanisms could operate there?
So there are other things going on. Obviously, I've focused basically on the splicing one. There's an unfolded protein response, which is a consequence of the fatty acid reorganization and really cellular reorganization. Oh, on that, no, so hypoxia or other things we have not tested, but there will be differences. Whether it is going through similar mechanisms or others, I don't know. I wouldn't think so.